sugar. And on and on. And this makes a chain. So this is one rung of the ladder, or one part of the chain, and it goes on and on. And I'll show you a picture of that here in a minute, but that's basically what DNA looks like in your body. So you have your deoxyribose, or simple sugar. It's 5-carbon sugar. Your phosphate group, and your nitrogenous bases, which are in DNA adenine, I believe thymine, cytosine, and guanine. These are the nitrogenous bases that make up DNA in your body. Moving on. This is just a quick overview of it, and then we'll get into a DNA PowerPoint that should elaborate some on what we're talking about. And I don't have a clock up here. I'm just going to use my phone for a timer. 4 minutes, thank you. So, DNA, the molecule of life. Again, as I stated earlier, there's trillions of cells in your body. Each cell contains 46 human chromosomes. That's 2 meters of DNA, like I said, something like this. 3 billion DNA subunits. The bases are A, T, C, and G. We're going to see how, in a few minutes, the actual recipe that's taken out of the cookbook, which is RNA, which is just ribose nucleic acid. We're going to see how that's a single strand, and thymine is not part of that. It's actually uracil, but I'll write this up here, but we'll get to that in a minute. Approximately 30,000 genes code for proteins that perform most of life's functions. So this is what I mean by DNA is double-stranded. So you have your backbone here, and then you have your nitrogenous bases. So your phosphate backbone, your nitrogenous bases that have to connect together. They can only connect in groups where they are similar, meaning A goes with T, and I'm sure some of you already know this, but so it doesn't matter how it's oriented, A has to go with T, because those are the only molecules that connect. A and T can only connect. Same with C and G. All right, now I'm going to show you a PowerPoint that is going to explain a little bit more about what we're talking about here with DNA. And real quickly, before we get to that, I'll show you. This is actually this is a representation of what DNA looks like in your body. Once it's unwound, obviously, like I say, when it's in this phase, it looks like a ball of string. But this is actually how it looks in your body. It's an interesting picture. Now if I can get back to my Dropbox. Okay, so here is our DNA PowerPoint. We're going to try to go through this as quickly as possible because I know time is moving quickly. So, here it is, DNA. And again, you see in this picture, you've got the base A matches up with the base T, the base G matches with C, and on, C to G, G to C, A to T, or T to A. And it doesn't show that, but it basically just flipped. So that's the only way it can connect together. That's the only thing that holds that string together is that hydrogen connection. So here we go. So what's DNA stand for? It's deoxyribonucleic acid. We covered that. The chemical substance is present in the nucleus of all cells in all living organisms. DNA controls all the chemical changes which take place in the cells. The kind of cell which is formed, whether it's muscle, blood, nerve, etc., is all controlled by DNA. So the DNA basically tells that cell what it's going to be. So when it packages up that chromosome, as we had here earlier, I'll redraw that if you forgot what a chromosome looks like. DNA in this chromosome will basically tell it what protein to code for. The kind of organism which is produced, whether it be a buttercup, whatever that is, a giraffe, a heron, a human, etc., is all controlled by DNA. It's a very large molecule made up of long chains of subunits. And like I said, if you laid out a strand of DNA, and again, there's 46 of these strands in your body, it's probably about that long. So that's a long strand. And you gotta think there's trillions of cells in your body. Each cell has 46 of these units. Each nucleotide is made up of a sugar, deoxyribose, a phosphate group, PO4, and an organic base. And that phosphate group is a phosphate connected to an oxygen connected by 4 hydrogens. So that's kind of how that works. Here's your ribose sugar. Again, it's pentose, and where they get that is there's 5 carbons. Okay, and off of each of these carbons, you'll have oxygen, and then you'll have hydrogens connected to that. It's just not shown in the picture. These are the most common organic bases. These are the only organic bases that make up DNA. Adenine, thymine, cytosine, guanine. Here again is what I illustrated on the board earlier, where you have one segment or one nucleotide. And these are joined together in succession in a chain, and that's where you get that long strand of DNA. It's a double strand of nucleotides, and again, I'll illustrate what this kind of looks like. It's helical, okay, so it kind of looks like this. So if you took a ladder and you were to twist it up, that's what DNA looks like. And again, this is in the prophase stage, or not the prophase, but the interphase stage. When it's unwound, this is what it looks like. Wound up, it actually looks like this. And I'm going to just breeze through this because this isn't actually the class. Here's a model of DNA. This is an example of what we're going to be doing as a project in the class. We're going to have DNA models, and you can choose from different sets of models, and then you can take them home. And this is going to be a 10-day unit project. You're to take this model, construct it, or a model like this, construct it, and then you're going to write an essay on DNA. And basically, what I want you to do is I want you to tell me about DNA as if I never heard of it through your essay, and that'll be due in Unit 10 with the day of the test. So again, before the cell divides, the DNA strands unwind and separate. And it goes through that process, and that's all called replication. Here's the separate strands that come together. Again, you have hydrogen that connects these, or as you can also see here, each strand builds up its partner by adding the appropriate nucleotides. Again, that's this connection here, A to T, C to G, T to A, and on and on. Okay, and rather than going through that whole PowerPoint for sake of time, we're going to go ahead and stop there. And I want to go ahead and move on to a assessment that I have here. Now, this was created for the students to do on their own, okay? But what I kind of adapted it for this, and the way I'm going to do that is just move on to the test, or to the assessment questions. And this has the answers, but in the form that we would use in the class, we would have the clickers for Turning Point, and then they could answer these questions. That's an assessment that I would use to see if the students retained the knowledge or the information about DNA. So RNA has all of the following except what? Deoxyribose. We didn't go much into RNA, but all that is just ribose nucleic acid. So it is missing an oxygen. That's what deoxy means. Deoxygenated ribose. So it's missing an oxygen molecule. the sugar found in DNA is deoxyribose, and then your next question is, how would this strand be produced from the following DNA strand? So if you've got this, we'll go with CC, TC, AT, GC. Now, again, this is the DNA strand. So when it's unwound and unzipped and broken into two strands, which would basically have your ladder cut through the middle, because there's an enzyme that breaks this apart. But you have these bases. Now this is what's called a semi-conservative model. These break off, and so you have an old strand of DNA, but then it builds a new strand